In today's episode, we dive into the intricate world of epigenetics with Dr. Johannes from the Technical University of Munich in Freising. His groundbreaking research bridges statistical genomics and plant biology, unraveling how heritable epigenetic variation influences complex traits beyond DNA sequence alone. How do methylation patterns shape phenotypic diversity? Can epigenetic modifications be predictably inherited? Join us as we decode the molecular signatures of gene regulation and their implications for evolutionary biology.
Welcome to the Epigenetics Podcast from Active Motif. Join host Dr. Stefan Dillinger for lively discussions with leading epigenetics researchers. Hear about their past experiments, what they're working on now, and what's coming next. You know their papers, now get to know them and discover the stories behind the science.
Hello and welcome to this episode of the Epigenetics Podcast. Today, I'm again not doing this via Zoom, this interview, but I traveled to Freising, to the TU Munich, and to the Institute of Plant Epigenomics to meet Johannes, who is our guest today.
Yeah, thank you.
A question I like to ask every guest to ease into the topic and to start off our podcast is, how did you become interested in biology in the first place and then in pursuing a career in science?
Well, good question. For me, it all started with popular science books. I was very intrigued by popular science books and the kinds of questions that they posed. So also at that point, reading a lot of philosophy books. And sort of was just juggling around a few ideas. And for me, science was the way to go. I was very interested in question of evolution and genetics. That's what got me into studying genetics, or rather statistical genetics in the United States. Yeah, and part of the questions that were interesting to me is how genetic architectures change over time, even within the lifetime of an organism, which led me kind of indirectly to epigenetic questions, because these are modifications of the DNA that contribute to genes being turned on and off. Had a chance then to move to France to get involved in a first population epigenetic study in plants. Is this what you want to know? Do you want to hear a bit about that, or you just want to?
Yeah, I mean, the next question would be how you ended up in your scientific life here at the TU.
Yeah, so I may as well continue that. And I got involved in that project that was spearheaded by Vincent Collot in Paris. Was there as a Marie Curie fellow my last year of PhD, and then also during a short postdoc time. And there we worked together on one of the first, like I said, population epigenetic studies in plants, where we try to show how heritable DNA methylation changes in plants can contribute to the heritability of complex traits. And after that, I moved to the Netherlands as a postdoc, where I actually continued working on these kinds of questions. Obtained a assistant professorship position there. And then later on was offered a Rudolf Mössbauer professorship here at the TUM IAS Institute for Advanced Study, which I took, and since 2022 I'm an associate professor here at the university.
So let's talk about your science that centers around chromatin-based mechanisms of genome regulation and epigenetic inheritance in plants. I want to start when you started your own lab, which was roughly around the year 2010, 2011, if I'm not mistaken. The first paper that came out of your lab was published in 2011, and there you extensively phenotyped 6,000 Arabidopsis plants with experimentally perturbed DNA methylomes. What did you find there?
Yeah, so what did we find there? So this was based on a population of so-called epigenetic recombinant lines that were essentially generated by Vincent Collot. The idea behind those plants is to perturb DNA methylation patterns in an otherwise isogenic background. And the way this was done is to mutating DDM1, which is a chromatin remodeler with an effect on DNA methylation. mutants of DDM1 have hypomethylated genomes. And so the idea was to essentially cross hypomethylated parent plant or wild-type plant and generate from that recombinant line individuals, which are isogenic, virtually isogenic at the DNA sequence level, but have varying or mosaic DNA methylomes. So we had profiled this material already early in growth chambers and in a greenhouse, and we showed that heritable DNA methylation variation was linked to important traits like flowering time, plant height, root length. And in that particular study that you mentioned, we also grew them out in the field and wanted to see how are they performing in the field. And again, we could show that there was a lot of heritable variation that we could detect. We also used that material at the time to do first estimates of the number of quantitative factors or QTL influencing these traits. We used not direct measurements of this, but we used so-called biometric estimates, which we later validated in later works with direct measurements.
So this was only like looking at DNA methylation, right?
This was only looking at DNA methylation, correct.
So then you tested the combined effect of removing sequence polymorphisms and repeat associated DNA methylation on the meiotic recombination landscape of an Arabidopsis mapping population. Can you talk about this study and what you found there?
Right. So so the study that you mentioned with these perturbed methylomes, this was all describing essentially phenotypic variation and doing biometrical approaches to show that induced heritable epigenetic variation contributes to complex traits. So at that point, we didn't have any direct measurements of these methylation changes, these epigenetic changes that had been induced. So the next step for us was to actually measure the methylomes of all these individuals. And we did that back in the days with tiling arrays. Whole genome bisulfite sequencing wasn't established that well yet. So we did all its measurements, and based on these measurements, we basically had now maps for over 100 of these individuals where we can study the inherited methylation patterns across these individuals. We wanted to actually use these maps to do QTL mapping, not in a traditional sense where you try to associate genetic variation with traits, but in this case epigenetic variations, we called epi-QTL mapping. But before that, we realized we could actually use that material to study the recombination landscape in this system. So there already was some evidence that DNA methylation may have something to do with recombination suppression, but there were no good methods to assess this at genome-wide scale, particularly in the isogenic system. But we could do exactly that. So we had material, all individuals were isogenic. We could study the recombination landscape, and we had sectors of these genomes that were completely hypomethylated. And we could ask, does the removal of DNA methylation, the DNA sequence variation in the system, substantially change the recombination landscape in the Arabidopsis? And what we had found is that in pericentromeric regions, where it was sort of thought that heavy DNA methylation leads to suppression of recombination, we found in our system, even in individuals that had hypomethylated pericentromeres, no significant impact on the recombination frequency in pericentromeric region. But what we did see is that the suppression of recombination was spread a little bit to the boundaries of the pericentromeric region. So somehow it did indirectly, but some mechanism we don't quite understand, affect the recombination and suppression in pericentromeric region. And then an additional trade-off for that was that the trade-off of having increased suppression at the boundaries of paracentromeric regions is that you have increased recombination frequency in chromosome arms.
Mm-hmm.
So that was our contribution with that. I mean, personally, I was never really deeply into the recombination field, but it was a natural step for us to go as we were generating these maps or QTL mapping that we could begin to explore these questions.
Mm-hmm. Okay, then I will move away maybe from this. Because the next publication, this was published in Science 2014, there you analyzed the population again of isogenic Arabidopsis lines that segregate experimentally induced DNA methylation changes at hundreds of regions across the genome.
Yeah.
I guess, can you talk about the study and your results there?
Exactly. So again, we're following the same research line that works with this population and this epigenetic recombinant line population. So what we had done there is to actually now do this QTL mapping. We'd use these genetics recombination maps that we had generated, and we tried to link heritable DNA methylation changes to complex traits. And these included flowering time and root length. And we could find multiple highly significant regions in the genome where differential methylation was linked to trait variation. We could show that these effects are heritable. We could show that selecting on specific traits, selecting on flowering type and root lengths, individuals that are extreme for these traits showed selection signatures at the level of DNA methylation. And we could also find that these QTL that we had detected explained essentially most of the heritable variation that we had estimated in earlier biometrical studies.
Mm-hmm.
So that was sort of the final demonstration for us that indeed inducing a loss of methylation across the genome can be inherited, in this case for 8-9 generations, and lead to heritable trait variation in important agriculture traits.
Did you also look at like the mechanisms in the heritability?
Yeah, so this indeed is still an outstanding question. What is actually causal in these regions? So, when you do this QTL mapping, you often end up with these large QTL confidence intervals that span often several megabases. You cannot really resolve them any further because you have no real recombinants in these regions. You can zoom in there and you can look at candidate genes, look at candidate differentially methylated regions, but nothing obvious had popped up at that point. I think the Colo lab was pursuing this a little bit further, but quite honestly, I don't know what happened to these follow-up studies.
But like 8 generations is quite a long time, right? For this to be heritable.
Well, that's not. I mean, yes, that's already a lot. But I think that the Colo lab is propagating these lines further for, I think, 30 generations. The last time I heard, and these induced changes are still stable.
No, that's very interesting.
Yes.
Next, you looked at forward epimutations and backward epimutations in Arabidopsis. To be honest, I've never heard of forward and backward mutations. So can you briefly introduce what those epimutations are and about the dynamics of those processes?
Right. So this is indeed the topic that we're pursuing intensively in my lab ever since then. And these are spontaneous epimutations that we define as stochastic gains and losses of DNA methylation that are heritable across cell divisions. And the first sort of quantitative study on this that we have done was this paper that I mentioned. Use of terms forward and backward, maybe a bit awkward, and since then they have been replaced by simply saying gain and loss rates, which is much more intuitive. So forward meant a stochastic gain of methylation. A cytosine is unmethylated, becomes methylated. And backward meant loss. Cytosine was methylated, becomes unmethylated stochastically. Yeah, so we have built basically on early studies in 2011 by the Weigel lab and Ecker lab, who had studied mutation accumulation lines in Arabidopsis and looked not necessarily at their genomes, but at their DNA methylomes, and wanted to just document if changes occur in the methylomes over these timescales. These mutation accumulation lines were 30 generations old. And we had sort of built on this to quantify the whole process, to find a way to really rigorously analyze or estimate these rates. We argued that having quantitative insights in these rates is important for the same reason that quantitative insights in the genetic mutation rates are important, because these are parameters that show up in population genetic models. They help you understand and predict genetic, and in our case, epigenetic diversity. Yeah, and we estimated these rates, and we found that they occur at a rate of 10 to the minus 4 per CG site per haploid genome per generation. And that was an interesting insight, because these rates are about 5 orders of magnitude higher than the genetic mutation rate. So
that means 1 in 10,000?
Mm-hmm.
Yeah.
So this means epigenetic diversity through this epimutational process accumulates much faster across generations than genetic mutations do. We also found that these rates are not uniformly distributed across the genome, but they show annotation-specific rates. So TEs show a lower rate of spontaneous epimutations than genes. So that also means, again, that epigenetic diversity at the DNA methylation level accumulates much faster in genes than in TEs. So if you have these rates per annotation, what you can do is basically ask, do they explain sort of broad patterns of DNA methylation diversity in natural populations? So if I slide along the genome and ask, what is the variation that I see in natural populations at DNA methylation level? Can we predict that from these epimutation rates? And there we found that we can do that, suggesting that these epimutations that we had estimated in mutation accumulation lines, which are essentially an artificial experimental system, can be informative about the kind of epigenetic variation that we see in natural populations.
So does that mean that there is a difference? Probably it does, but does that mean there is a difference between repairing the genome, so damage on the genome, and repairing those differences or those defects in DNA methylation? So maybe, did you look at that, or can you speculate?
Yeah, so one small detail is that, as you know, maybe you don't, maybe you know, I don't know, but DNA methylation in plants does not occur only in CG contexts, but also in non-CG contexts. So they're quite distinct in that regard from animals, from mammals. And what we found there, if you track the accumulation of spontaneous epimutations in these different contexts, you see that they accumulate over generations in CG contexts, but not in non-CG.
Mm-hmm.
So things are heritable in a CG context and essentially non-heritable in a non-CG context. There's some biology behind why that should be. So we focused mainly then subsequently on CG context. We were interested in things that inherited across generations and build up across generations in these genomes. So CG methylation in plants is maintained by a specific methyltransferase, and what it does is basically maintains methylation at cell division. So when DNA is synthesized, it basically looks at the parental strand and sees that it's methylated, and then it deposits methylation in a newly synthesized daughter strand. So it's what it uses a template in a sense to do that. Now, so this is an enzymatic activity, and mistakes can occur in this enzyme. where it fails to deposit methylation, and this is a mechanism for us how loss can occur. Once loss has occurred at the CG site, it cannot get reestablished at sites that are only targeted by a maintenance methyltransferase. The only exception is if there's a de novo pathway targeting these CG sites that can deposit de novo methylation. But there are many, many sites where this is not the case. So when things are lost there, they're then stably inherited and cannot be recovered.
Okay.
So that speaks to your repair. It cannot repair it in a sense. Gain is a little bit more tricky at these sites. We don't quite understand where gain is coming from. There is some evidence that the same methyltransferase that I've just mentioned, that's responsible for maintenance of CG methylation, also has this occasional de novo function. It goes to sites and somehow makes a mistake in depositing methylation where it shouldn't. Yes. Exactly. So there is indeed some research to try to figure out if this has anything with the DNA repair. Are these events? Is there maybe another explanation for these gains and losses of methylation, possibly linked to DNA repair? But this is ongoing research, and I cannot really say too much about that.
Okay, what you also looked at was epimutation hotspots and the molecular properties of those. Can you talk about what you did there?
Right. So we kind of went back to this early paper where we studied epimutations we found and not uniformly distributed across the genome. Back in the days, we did this at a very broad resolution, and we only did this. So basically, scan across the genome in like megabase windows, and we found that rates are higher at this resolution. And we also did this in an annotation-specific fashion, so where you looked at annotated DNA elements and their different epimutation rates. But we thought we could do a little bit better. We can look at a higher resolution maybe, and instead of just looking at annotations like genes and TEs and promoters and what have you, we look at chromatin states. There was already some evidence that there was some association between specific chromatin states and the ability to maintain DNA methylation with higher fidelity or not. So we went to a database that was generated not just for Arabidopsis, but I think also for maize, that annotated the genome in terms of different chromatin states. And chromatin states in this sense here are sort of combinatorial patterns of different histone modifications or other chromatin modifications. Yeah. So we basically had this annotated Arabidopsis genome. We just estimated rates for different chromatin states, and we found that the rates were different for different chromatin states, and we could use basically the abundance of specific chromatin states in a region to make predictions about epigenetic diversity over generations via their link to differential epimutation rates. Yeah, that worked quite well, and we found that there are some chromatin states indeed that sort of are associated with epimutation hotspots. They showed rates that were 3 orders of magnitude higher than other places in the genome. And when we sort of explored these chromatin states, we found that they were essentially mapping to so-called gene body methylated genes, which was quite interesting because gene body methylated genes have been defined as evolutionary conserved housekeeping genes that display high CG methylation, consistently high CG methylation, and essentially no non-CG methylation. And it had been shown in previous research that actually the DNA methylation levels, at least, so the average methylation in these genes pretty much conserved over evolutionary timescales across very diverse plant species. So it was surprising to see that actually in those highly conserved genes, you would have a higher rate of making mistakes and maintaining CG methylation at individual sites. Yeah.
What kind of consequences would this have if you have more mutations in those genes? It's just expression-based differences then, probably. If you have more methylation in the gene body, it would be less expression, right?
Say it again.
If you have more methylation in the gene bodies, you would have lower expression.
Yeah,
right?
no. So so there's a whole discussion about the function of gene body methylation. Nobody really exactly knows what it's doing, but there's no clear link to expression at all. So what you find it in moderately expressed genes, but there's no sort of clear linear relationship between methylation levels.
Also no fine tuning.
Yeah, it could be fine-tuning, stabilizing expression. These are housekeeping genes that have to be on all the time. So there's some evidence that this may be fine-tuning. Some people say that it helps in splicing. There's several explanations. None of them have really completely panned out yet. So we don't really know what it's doing there. Yeah.
Okay. So something that was already part of some episodes on this podcast is epigenetic clocks.
Mm-hmm.
And they have been around for some years now in mammals, but I was not aware that this was also done in plants.
Mm-hmm.
But you worked on an evolutionary epigenetic clock in plants. Can you talk about this study and what this can then be used for?
Right. So these epigenetic clocks in mammals are remarkable. There's based on the observation that DNA methylation at specific loci is highly correlated with age. And these, using just methylation measurements for an individual, you can predict what their chronological age is. And depending on how these clocks are calibrated, in some cases also biological age. That's an interesting observation. One of the things to know about these clocks is that they're based on a statistical model that takes population level data of measured DNA methylomes from individuals. It basically tries to find in the first step specific loci whose methylation levels are predictive of age. So you start out, they often in the mammalian field, in humans, they work with these large arrays that sample 400,000 sites, maybe 800,000 sites, mostly in CPG islands. That's the starting point, and then they do a model prediction and end up with, depends on which kind of clock it is, it can range from like 10 to several hundred of loci that are highly predictive, collectively highly predictive. The interesting thing about these clocks is that once you have them calibrated, you know what these loci are, you can go to any other individual and just measure these loci and the methylation level that this individual has at these loci should be predictive of age. This in turn implies that the changes that occur at these loci are highly systematic. They are just always targeting these loci, some systematic byproduct of aging. Yeah. So, but we came from another perspective with our plant work. We worked on stochastic changes. They don't always occur in the same place. They're occurring essentially all over the genome. We can never predict in one generation to another, in one individual to another, that a change happens at specific sites. But nonetheless, we thought we could maybe use these types of stochastic changes as a way to quantify an evolutionary clock. In the evolutionary field, molecular clocks are routinely used to estimate when two species or two lineages share the most recent common ancestor. And the way this is done is usually you look at DNA sequence divergence between the lineages or species. And if you know the mutation rate at neutral sites, you can estimate when they must have shared the most recent common ancestor. These classical clocks rely on DNA mutations. We all know that DNA mutations are very rare. They occur in most species around 10 to the -9 per site per generation per haplogenome. So if you go to timescales that are very recent, say, a few decades, a few hundred years, you often don't have enough mutations that have accumulated into lineages to confidently predict when they share the most recent common ancestor. So we thought with our epimutations, we have two properties that are quite new. One is that they occur at a rate that's 5 orders of magnitude faster, so the clock ticks much faster. And the other property is they're heritable. So we could actually, in principle, use that information to define an evolutionary clock. And that's what we did in that study. So we looked at material, at mutation accumulation lines that are 30 generations old. We calibrated an epimutation-based clock, and we could estimate that indeed these individuals share the most recent common ancestor 30 generations ago. We tried another non-model species, which is Stostomarinus, the seagrass. It's clonally propagated. And similarly, based on experimental data of seagrass clones that had been maintained for 17 years in tanks, we could calibrate and estimate that they indeed showed their most recent common ancestor 17 years ago. So we had good evidence these clocks work quite well in an area of evolutionary time where DNA-based clocks would not work well. And we could also demonstrate that experimentally, that DNA mutations just have too little information over these timescales. So applications are indeed to explore sort of the diversity that mechanisms that generate diversity over very recent timescales of decades to centuries.
Yeah, that's interesting, and also a little bit different than what mammalian clocks are then used
exactly.
for. Yeah?
That said, these types of mammalian clocks that have this sort of systematic component to it, they also exist in plants. And this has been demonstrated in trees in at least 2 studies, and our group is also working on this right now.
Okay.
So we're really training these classical mammalian clocks in trees to see if we can define an aging clock.
Yeah, that feeds into my next question. Imagine you are due to submitting a grant proposal tomorrow. What would you have written into this?
Tomorrow? Oh, I submitted one yesterday.
There you go.
Yeah, no, this clock, this clock stuff is really interesting us right now. We're working on projects, and obviously determining age in trees is an interesting aspect, because it's not easy actually to do that. To this accurately, you have to core into the tree. Not only is that very laborious, sometimes you're not allowed to do it with protected trees. Sometimes trees don't have tree rings. You cannot count anything. And when you talk to the tree people that do the coring, you have to really make sure that you do this correctly. Otherwise, you even have there an inaccurate estimate of the actual age. So we thought that was a very nice, non-invasive strategy to do this.
So it would be enough to take leaves, for example?
We do this on cambium. It may work on leaves, but we do it on cambium. Yeah, that is just the layer under the bark. Yeah. So preliminary results confirm that this probably works, but this is indeed something that we're now trying to dig into. Another aspect that is also important for the mammalian field in these clocks is that once you have a calibrated clock that predicts chronological age, it also captures an aspect of biological age. And sometimes you see situations where the DNA methylation clock tells you that you look like you're a younger person, or you look like you're an older person. So if the clock tells you look like an older person, then your actual chronological age is called accelerated aging. And that has been shown to be correlated with early mortality, diseases, etc. So in the biomedical field, this has become a big thing. For trees, this should also be interesting, because it's not always so obvious to tell the vitality state of a tree. So our hope is to calibrate these clocks and then also use them as a screening tool to assess the vitality of individual trees, or maybe plots of forest that have been, say, hit by drought or some other pests. It might offer alternatives for forest management.
Yeah, that's also very useful in the future. So for the last 35 minutes, we have been on a journey through your scientific career. Did we miss something important, or would you like to add something?
No, that's we probably missed one part where we're really interested in trees. All this epimutation stuff started looking across generations, but at this point we're actually quite interested to look at the accumulation of epimutations within an individual, so within a plant, and
so in different parts of.
That's right. So it turns out that trees are actually super cool models to address that, because they have done the epimutation accumulation, or also mutation accumulation for you. They've been around for 200, 300 years. Accumulate these things. All you have to do is take measurements now, and then you have basically the data that you need to work with.
How do you grow those trees? Or where do we get the sapis from?
Well, I mean, they grow in the forest.
Yeah, of course.
I also work together with people that work in fruit trees, so they grow them in orchards. Yeah, and yeah, so there are some interesting questions in trees that border on cell biology. So one of the interesting things there is that we know that a lot of these mutations that then spread throughout structures of the tree, or epimutations that spread, their cell lineages originate in the stem cell center of the plant. And once you know that, then you suddenly have to know how, what does the stem cell center look like? How are the cells set aside that make lineages to produce lateral organs and things like that? So all these parameters suddenly become important to understand how somatic mutations and epimutations accumulate in an individual. And this is something that we're pursuing from an experimental standpoint, but also from a modeling perspective.
Okay. So my last question would be, what is your personal definition of epigenetics?
My personal definition of epigenetics is there used to be a YouTube video a long time ago where they asked some real experts on their definition, and they're always stumped by what it could be, and they were quite divergent. So these are heritable changes in a chromosome that are not due to DNA sequence changes. I deliberately don't put a phenotypic component in there, which is found in many of the definitions, that it should lead to heritable change in gene expression, simply because a lot of the heritable epimutations that we study have no phenotypic consequences, so they're neutral, but nonetheless accumulate and show epigenetic inheritance.
Okay. That's a very precise and concise definition. That's great. So thank you, Frank, for your time and for being on the show.
Yeah, thank you.
Thanks for listening to this episode of the Epigenetics Podcast from Active Motif. We hope you enjoyed it. You can find all the mentioned references in the show notes. Please rate, review, and subscribe to our podcast on your favorite podcast platform so you never miss an episode. We'd love to hear from you, so please send us your feedback on Twitter, Facebook, LinkedIn, or via email at podcast@activemotif.com, and we'll give you a shout out in a future episode. For more great epigenetics content, check out the Active Motif blog at activemotif.com/blog. Thanks for listening, and stay tuned.